Hello, welcome to my channel Yashoda Krishna 1392. Today we are going to learn drugs from natural source in the treatment of cancer, mainly concerning to anti-tumor antibiotics. Anti-tumor antibiotics. So what are the drugs? So let us learn some introductory part which are concerning to natural source. The drugs which are obtained from the natural source and which are approved are from the microbial source and plant source. Nowadays, some importance is given for the drugs which are obtained from the corals, from the plants, taxines, Taxol, paclitaxel, those drugs, vinca alkalides, vincristine, vinblastine, phodophyllotoxin, camptothecins. These are the some of the drugs which are approved and marketed in treatment of cancer. What else? It is in the introductory phase, a developing phase, ilotorobin, BMS 247550, sarcodictines, dictines. So these are the drugs which are in phase to different phases of clinical trials in the treatment of cancer. So drugs which are obtained from the microorganisms are called as anti-tumor antibiotics. Anti-tumor antibiotics. Let us learn what are various drugs which are available. First, let us learn the classification of drugs. The drugs which are available are, you can classify based upon the chemical structure. Anthracyclines. In anthracycline, doxorubicin, daunorubicin, doxorubicin, daunorubicin, epirubicin, idorubicin, mitoxantrone. Mitoxantrone. All these are from the anthracyclines. These are from the anthracyclines. One more class of drugs belongs to anti-tumor antibiotics are actinomycins or chromomycin. They are colored substances. That's why another name for that is chromomycin, that is dactinomycin and pilcamycin. Miscellaneous drugs, the structures are they're different. That's why we are classifying them as miscellaneous. Examples are mitoxantrone, mitomycin, bleomycin. All these drugs, all these drugs are cell, non-cell cycle specific drugs. That they doesn't act at the uh, growth of the cell. They act at different phases of the cell cycle. Whereas bleomycin, acts at G2 phase of the cell cycle. G2 phase. Specifically, it acts at the G2 phase of the cell cycle, whereas other drugs are non-cell cycle phase specific drugs. Cell specific drugs. And all these drugs are obtained from the nature, from the soil, that is the fungus, from the fungus of soil called Streptomyces species. Streptomyces. So these are natural ones. So from the soil they are obtained. So that's why we are telling natural ones. Anti-tumor antibiotics. So these are the drugs. Based on the chemical structure, we classify them. Now let us learn what are various mechanisms how these drugs act. So these drugs act by intercalation. Number one mechanism is intercalation process. The main chemical property which is responsible for intercalation process is that they are flat in structure. The ring is flat in structure. So the ring is flat. The rings are flat in structure, especially aromatic portion. This aromatic portion is responsible for slipping between two DNA molecules. Here also, this phenoxazine ring is aromatic in nature So it slips between DNA molecule with the slips between DNA molecule, just like this. You can see it here. See, this is aromatic. This is a aromatic ring. Now this aromatic ring is, you can see it here. This is the uh, this is a doxyrubicin. You can see it. This is one strand of DNA. This is another strand of DNA which slipped between the two DNA molecules and causing intercalation process. Intercalation process. So that is the main important process which these drugs act. Number one is intercalation process. Next, after binding, after entering into the sleep, they cause strand breaking. They cause strand breakage by free radical generation. We will study that. The free radical generation. Next one is enzymatic inhibition, that is by blocking topoisomerase 2. Topoisomerase 2. All the drugs, whatever we discussed in previous slide, will act by number one, number two, number three. Alkylation is one special action, mechanism of action for mitomycin C. Just like alkylating agents, um, cyclophosphamide, just like that, mitomycin C will act. Apart from these three, it also blocks it also generates free radicals. It also blocks topoisomerase. It also acts by intercalation. Along that, it has alkylation process. Alkylation process. So this is the way how they act. So what are the steps? Various steps involved in intercalation. So as I told in previous slide, they contain aromatic ring, which is planar in structure, planar aromatic ring, or heteroaromatic rings, as we have seen this drug, actinomycins. In actinomycin, what happens is that in actinomycin, you see, there is a hetero aromatic ring structure. It is a hetero ring is present. So because of this aromatic nature, aromatic nature, they slip into the double-stranded DNA helical structure and distort its structure. So because of slipping into DNA molecule, structure of DNA will be stopped, will be distorted. So that's why these drugs are intercalator. Then the double bonds in the intercalator, there are, as we told, that planar aromatic means sp2 hybridization, nothing but double bonds, imparts planarity and also responsible for intercalation. Then the sugars which are present in it, the sugars has amino group and hydroxyl group, which also they also form by hydrogen bonding, and cationic ionic bonding. Ionic bonding, they intercalate with the phosphates and sugars present in the DNA molecule. This aromatic ring will slip between the DNA base pairs. Now the sugar molecules will form hydrogen bonding. The amino sugar hydroxyl group present on intercalator form ionic and hydrogen bonding with the anionic phosphate on the base pairs. So this is how intercalation. There are many interaction between the DNA and DNA. Now this is the planar structure, the aromatic rings. These three aromatic rings by hydrophobic interaction, the sugar molecule and these hydroxyl groups. You see the hydroxyl groups which are present on the molecule will form hydrogen bonding, and this the sugar molecule has amino group which becomes positive during in the physiological pH. and form ionic bonding with the phosphate present in the DNA molecule. So tight bonding is present between the drug, for example, doxorubicin, then with the DNA molecule. That is one. Next one, you can see it one more way of diagrammatic representation. Now you can see this is the drug molecule. Now this is one DNA pair. This is also this is one strand. This is one strand. Now in between the DNA strand, now our drug is attached. Where it is attached. Now when it is attached, what happens is that there is overall change in the shape of the DNA molecule, which causes bend or kick or cut of the DNA, resulting in shape distortion of the DNA. So this is one. The intercalation also prevent DNA unwinding by blocking the activity of topoisomerase or DNA gyrase enzyme. So when there is no unwinding, so DNA has to unwind for the replication process. When there is no unwinding, there is no further DNA process. So thereby, cancer cell will die. Cancer cell will die. So the free radical mechanism is that now here the drug is in quinone form. The C-ring is called as quinone form. Quinone form. This is the C. This is called as quinone form. Now quinone form, what happens will react with NADPH. NADPH will undergo oxidation process, will undergo oxidation, now it is gaining one electron. You can see it now, it is semi-quinone form. It is called a semi-quinone form of, this is a doxorubicin structure. We can't tell anything because we have given R. So anthraquinone glycosides will react with NADPH to form quinone form. Now quinone form will convert again to uh, semi-quinone form. Semi-quinone form will convert to quinone form by reacting with oxygen. Now, when oxygen reacts, what happens? There is formation of superoxide. Oxygen minus one electron. Oxygen minus one electron. This is called as superoxide. When superoxide reacts with hydrogen with the help of SOD, superoxide dimutase, you are getting hydrogen peroxide. Hydrogen peroxide. Now, this hydrogen peroxide will react with the ferric form, will react with the ferrous form of iron, and which converts to hydroxyl ion. All these are free radical, that is hydroxyl ion free radical, hydrogen peroxide, and superoxide are free radicals. These free radicals will damage the DNA molecule, So this is how these drugs act. So they intercalate, then they block topoisomerase enzyme, they produce free radicals which damage the DNA molecule, and these free radicals are also responsible for some side effects, especially cardiovascular side effects with these drugs. So let us see some of the questions in this section what we learned. Now, which of the following drug has anthraquinone anthracycline structure? So which of these? Mitomycin, bleomycin, pilcamycin, doxorubicin. Which of these drugs has anthracycline structure? The one is answer is doxorubicin. Doxorubicin is the one which has anthraquinone anthracycline structure. Which of the what are various interactions? That means bonding exists between anti-tumor antibiotics and DNA. Van der Waal interactions, ionic bond, covalent bond, or all the above. Try to recollect. It is van der Waal and ionic bonding. Van der Waal is the aromatic ring. Ionic bonding is because of what is that? Amino sugars present in the drugs. We didn't give what is that? Hydrogen bonding. If there is hydrogen bonding, then options could be all the above. But instead of that, we have given covalent bonding. Now, what is the source of uh, sacrodictins, uh, dictins, and illotobin? These two are not from the microorganism. Microorganisms are anthracyclines, phenoxanthines. So what are the drugs? It is about from the corals. They're also not from the synthetic. They are from the corals. Thank you. These are the references which we got. This is from the William Foy, Wilson and Giswold, chapter 37, Wilson and Giswold, chapter 30, chapter 10, then Graham Patrick, chapter 21. The page numbers are given. Please refer them. If you have any doubt, please comment. Thank you very much.